So thank you very much, Minigi-san, for your introduction. And good morning, everybody. I'm Tadashi Kajita, and today I'm going to talk about eDNA biodiversity assessment in mangrove ecosystem. Then, although I'm the speaker from our big group of collaboration, but only the representing speakers, then I'm going to introduce the basics or the framework for this international collaboration, and you will see a more detailed report from the next following three speakers, like Alison, Habib, and Venus here. Then we also have other collaborators on this floor, like Chen and Chen from Malaysia, Gustavo Mori from Brazil, and Ichika from Thailand. So if you have any questions for each cases of study, please speak to them in the coffee break. Okay. Then today, what I talk is about mangrove. Then the distribution of mangrove like this, the coastal region, which is colored, is the distribution area. So that you can see this is global distribution. So around the world in the subtropical region or tropical region, you can find the mangrove mostly. But as you see, the distribution is very thin and only along the coastal side. So this is, the mass of the mangrove is not so very big, like only less than 1% of total tropical forest. So still rare, I can say, or vulnerable. No, not so any rare ecosystem in the world. Then, as you see, here is a showcase of mangrove landscape in Brazil, Senegal, Malaysia, Japan, in Urimote. I'm not sure you can identify which is which. Then from the top here, this is Brazil, Senegal, Malaysia, Urimote, Japan again, and Brazil. Everything has slightly different, and I can identify which one which very clearly, but maybe for you, this is very, same. So everything one thing, mangrove would be same in the world, but not actually like that. Then the mangrove is quite important for the human being, especially for the human well-being. It is written here. This is a report from IUCN and UN. Then the ecosystem service, so-called, is for livelihood, wood, and coastal protection, water filtration, tourism, and fishery, and climate regulation. So if the report estimated the value of mangrove in money, then it was 800 billion annually. So that is something similar to the national yearly annual budget of Japan. So quite a very high value. But however, mangroves are under threat from human activities. So the point is here, now encircled in red, 35% of mangrove has already get lost in between 1980 to 2000. So yeah, actually we already lost most of quarter of the world mangrove from the world. Then the reason was lodging, agriculture, aquaculture, and pollution, coastal development, and climate change. Then after that, after 2020, we still had more recent statistics based on the satellite imaging. Then the red region on the map is the loss is still ongoing. Then the main driver here is a bit difficult to see, but here is land use change for the agriculture and aquaculture and oil palm plantation. So oil palm plantation is now main driver in the Southeast Asia to degrade the mangroves. But at the same time, we lose mangrove, we are losing the biodiversity in mangrove. So you see, we have quite a lot of diversity of living fauna in the mangrove, but it is getting lost when we lose mangrove. But how we lose them is a question. So many people are living in the mangrove country is aware of we are losing something biodiversity in mangrove, but how we lost biodiversity is a matter. Then in 2017, we have one review paper on the report of biodiversity estimates around the world. This is a table written like this in the geographic region, and in the column is like organism from plant, fish, insect, crustacean, blah, blah, blah. But you see, even though we have quite a lot of information for plant, because we can visit there, we can identify them and count them. But for other animals, others, we cannot know how many. And all the red part is written ND. That means no data. So even though the fish and the crustacean, which is relatively known, we still find some country has no data yet. And also the estimated number is very different by country. So it is not quite correct, I think. Okay? So this is a very big challenge and knowledge gap for us and for the people living in mangrove country. So we need more study to reveal the faunal diversity, how it is we still have, and how it is getting lost. So, but there's a very big challenge for the global observation because mangrove distribute around the world. So it's quite difficult to do the field work and also conventional taxonomic works for this very wide region. So like wide-scale observation difficult, and expert who can study everywhere is very limited. And for that study, very, very long time and effort is necessary. So that using the conventional method to rapid acquisition of mangrove biodiversity knowledge on a global scale is very, very difficult. So we thought about the two breakthroughs. One is international collaboration. So to reveal the biodiversity and the present condition, no single one country cannot do that. So that international collaboration is quite necessary. Another one is, as Kondo-san from yesterday, he pointed out, eDNA technology. It can be the very, very good tools to do that. So, okay, I start from the first breakthrough of international collaboration. So I'm not a fish taxonomist. I started my study from the plant taxonomy. Then I had some experience in the mangrove science for the genetic part. So that using that network as a basis, we started this collaboration. Then this is supported by JSPS Core-to-Core program for the Asia-Africa framework. So this is quite a unique program to let us network in the Asia and African region to tackle for the same issue, same question and problem by using the power of the network. So at first, we make this network with 9 nations in Asia, Africa, and some supporting country from the other part of the world. Then in this collaboration network, through the seminar, workshop, field training, and lab training, they are the ones that the JSPS Core-to-Core support, not actually the main research, but for networking through these activities. Then raising capacity building in each country, especially for younger generation, is the most important part. So we work together towards the same objective. So to use the eDNA Metabarcoding method to assess the biodiversity around the world. So this is our first start in 2020. Then this work went quite well. Even though we get quite a big influence from the COVID pandemic in the first 2 years, then we couldn't meet each other. So only the first 2 years of online meeting and online activities. But yeah, we did very well. Then we could expand our network and also can get support from JST. That is two main big projects. That is eAsia and AJ Core. So eAsia for the supporting the collaboration. In this case, research, not only the networking, but research with Philippines and Indonesia. Then for AJ Core is to support the research with South Africa and Senegal. So all the 4 members are from the JSPS Core-to-Core members. So using the JSPS Core-to-Core as a basic vessel, so we started and expanded our project. And luckily in 2024, we can get the second round of JSPS Core-to-Core in collaboration with Kondo-san, Minegi-san, and all the members of the previous Core-to-Core. And now it is supported by Tohoku University's Nature Positive and Ocean Short, and now expanding for ANEMONE Global. Then now we are now communicating with Aubon as we ANEMONE Global get the endorsement for the Ocean Decade. Then other international project is also involved. So the framework between the difference of JSPS Core-to-Core and ANEMONE Global is like this. So we established this is a second phase of the framework. We have, I think, 12 institutions from the, I think, 9 to 10 countries. Then this collaboration framework was moved to ANEMONE Global. So most of all the members was joined to ANEMONE Global and make the basic framework for international collaboration established especially for the West Pac regions. Then it was supported by these important funds, and now get a Union Ocean Decade endorsement. Then now, this time, we just submit a proposal to get another endorsement for the mangrove project, because the aim is slightly different, because we are more specific to mangrove. So now we are trying to get the endorsement from Ocean Decade for mangrove project as well. Okay, then second breakthrough is eDNA-based mangrove biodiversity studies. So I think most of you here know what is the eDNA studies. Then I will quite simply explain this part. So this is like so-called My Fish workflow established by Dr. Mia. Then the most important part for us is this part, water sampling filtration. So in some cases, in some occasions, if this is easy cases, we can finish the sampling in just 30 minutes in the field. Just collect water, filter it, and you don't have to have an expert. If you know the field very well, you can just visit them there and collect the appropriate water and filter it. Then the sample can be shipped to the laboratory and used for the extraction, library preparation, and sequencing. So in the laboratory, you can get all the data, very massive data in parallel. So the most important part for us is the quite easy, non-invasive method and high throughput, and the data obtained at first is very objective because they are sequence data, comparative around the place to place or around the world. Then in this project of JSPS Core-to-Core, we're using MyFish and MyDeca, two markers. MyFish is for fish, using the 12S ribosomal RNA or mitochondrial DNA. And another one, MyDeca, is 16S. Both of them are quite short, but it's easy to amplify using the eDNA samples. Then, as I said to you, from the first of this project was 2020 under COVID. So what we did is at first, we prepared the video manual because all our participants can do their field work by themselves. So by using, this is a field in Iriomote. So Iriomote is very easy. Just walk into the mangrove, scoop water, filter it. So we thought it's quite easy to do this. And other countries can do that, but actually, like this. So here's Alison, and she did this field work 2021. Then when she went to the field in Malaysia, the water is like that, like coffee, okay? And milk coffee. Then she called me from the field by phone. Yeah, I cannot filter it quite much. Is it okay to use coffee filter to filtration? I had no idea, actually. So, but I said, yeah, maybe it's okay. So actually, she's doing here, using coffee filter, pre-filtration, and even after pre-filtration, the sediment is like that. The water volume, she could filter it very, very low, like 300 to 400 per slivix. And after that, we can visit Malaysia and think about the way, and we started to use a corking gun, which helped us more. Then using this one, maybe 500 milliliter in this environment can be 400 to 500. But then it was used in Senegal. So in this field of Senegal, it's very clean water, so quite easy to filtrate pump. But in this one, in the northern part, very turbid because of the sediment. So here we can only filter the 200 milliliter per slivix. We have to use 5 slivix, and it took cost and time in the field. So the method is anymore very easy, not so quick. So we started to use another technology, which is called quick conc technology. In this case, we can just put the even turbid water and like shake it and filtrate it using the this like pouch, plastic pouch. So the result is we still have comparable data for this field, but in Japan case, the data is okay. So that maybe we are going to use this one more in the world mangrove field. Okay. Then next one. Sorry about that. Okay. Then maybe I better to skip the workflow for the data analysis. Slightly different from the ANEMONE global because we do not use a semi-quantitative method. So this is just a real number can be obtained. Then this is the important comparison by country to countries. So we compare the data with the BLAST reference data. So the gray part is non-similar sequence data with a BLAST database. So in Japan case, very small. So almost 90% is similar. We have similar data in BLAST. But for other countries, 40 to 50% is not similar in the BLAST database data. So this means reference data, which is the most important part to taxonomic assignment, is still very low in these countries. So this is the one point we have to invest more. So in many countries like Habib and Venus is already started working on the development of a reference library, reference databases, I mean. Then just a quick view of our data. Now we have compared this number of data, countries for mangrove data. Then, yeah, something like this. So in the Southeast Asian region, you will see the distribution of so-called representative fish species in this mangrove area. So some of them are quite popular for you. For example, tepo, tohoset, and other ones is maybe tilapia is also here. They are, of course, the introduced species. Okay. Then another important outcome of this one is like capacity building. So so far we held 13 international workshops, including 2 international symposiums in Iriomote and other parts of the world. So the most recent one is in Senegal. Suyama-san and I visited there to help them to establish the laboratory over there. Then this is a regular workshop held in Iriomote. So we have 2 courses of maybe workshops. One is basic courses. So there in 7 days, participants can study how to do the field sampling, DNA extraction, and library preparation, and sequencing. Because we have the in-house sequencer, then we can do the data analysis. So now, after the visit to Kazusa the day before, we are thinking that if Kazusa can help us to hold this kind of workshop in Kazusa, so maybe we will talk with Yamaka-san later. Okay. Then another one is only one workshop is not quite enough, because after the first training, those younger generation come back to their own countries and do their own research. That time, how to conduct their own research is another matter. So that we have more like a custom-made workshop in Iriomote, invite those younger researchers, especially who have the samples to work, and they come back to Iriomote and work together there to get the data and to maybe get a publication. So far, we involved about from 9 countries, 13 graduate students, 2 master course students, and 11 PhD students. And some of them have already finished their PhD and masters. Then we are continuing these activities. This is so-called like building the next generation of mangrove biodiversity research. So I think this is the end of my talk. Sorry to talk long. So anyway. I think the next 3 speakers will talk more in their real studies. Then, if you have any question and you need my support to answer, then I can also help them to answer. Thank you very much.
Great, thank you so much. So, if you have quick questions, I can take 1 or 2. If you have, can you raise your hands? If you have any. No? No questions? It's okay? Mia-san.
Thank you very much for the talk. It's more about comment than the question, but I'm really impressed and very touched about the distribution of your network. I think eDNA has been applied a lot in like Europe, US, Japan, in like those developed countries, but then not so much in other countries. But your network is really filling the gap. So I'm really touched about it. And for me, like I'm working in Australia, and then, how can we really support in that kind of initiative? Like I would really like to talk with you, and like for us, like we're always willing to do that, but maybe we don't have connections, we don't know how to start with. So if you could help us in that way, I would love to do that.
Thank you very much. Very good comment. Then, as you said, maybe why we can make this network was we have a long-standing collaboration from the time of the plant studies. Like Alison, so we know each other while she was a student at that time. Even Gustav, just after finishing the PhD, like that. So maybe, then how answer to your question is we already have something like Aubon. So we belong to help Aubon. So if Aubon expands their network using also our network, so I think you can expand, or Australian people also work with other people through Aubon something. So maybe the role of Aubon can be the hub like this. Yeah, I think.
Yeah, that's true. Thank you very much.